Dear children, let me introduce to you a new advancement in technology, that is tissue culture. Tissue culture is a method of asexual reproduction in which we can propagate a large number of plants from a single parent in a laboratory under sterile conditions. Sterile conditions means which is free from microbial contamination. So that's called as sterile condition, that is microbial free conditions called as sterile condition. And what are the advantages? Why do we prefer tissue culture? It is because it helps to propagate a large number of plants from a single parent. Exact copies of the parent are produced from a single parent in a very short duration of time under sterile condition. It also helps to produce exact copies of the parent plant in the absence of seeds. Not only in the absence of seeds, it also helps to produce new plants from those seeds which have very low rate of germination or growth rate. For example, orchids, nepenthes, all these are propagated through tissue culture. Another great advantage of tissue culture is that it has to propagate disease-free plants or virus-free plants from an infected plant. So that is a very great advantage of tissue culture. Now let us understand the method. What is the procedure which is followed in tissue culture? The separating tissues or cells are removed from the growing tips of the plant. We know what are growing tips. Those which have meristems. Meristems means those cells which has the capacity to divide. So the growing tips have meristems. And from these meristematic tissue, the separating cells are removed. And they are placed in a medium, in an artificial medium in the laboratory. And in the artificial medium, it starts to proliferate because meristems has a capacity to divide. It starts to proliferate to form a mass of cells, and this mass of cell is called as a callus. Once a callus is formed, it is transferred into another medium which is having growth hormones. And inside this growth hormone containing medium, the callus undergoes growth and differentiation to form small plantlets. And all this is taking place in the laboratory. Once the plantlets are produced, it is removed from the laboratory and placed into the soil so that it can face a natural environment, and there it matures into a new plant. So this is the procedure which we follow in tissue culture. So the growing tip is taken, and from there the separating tissue or the removing tissue is taken out and placed in an artificial medium. And once it is placed in an artificial medium, it grows into a mass of cells called as callus. Then the callus is again transferred into another medium containing hormones, where it undergoes growth and differentiation, and it grows into a plantlet, and the plantlet is transferred into the soil. And this is a very important technique to grow a large number of ornamental plants. So hope tissue culture and the procedure and its advantages are very clear to you. Now that we have finished fission, fragmentation, regeneration, budding, vegetative propagation, tissue culture, all these methods are over. So we are going to get into the last type of asexual reproduction, that is spore formation. Dear children, if you buy bread and you forget to keep it in the refrigerator, after a few days, you can see a cottony mass appearing on its surface. And if it is left over as such, after a few days, we can see a grayish colored mass covering the entire surface of the bread. And this is called as bread mold. It's a fungi called as bread mold, and its scientific name is rhizopus. The structure of rhizopus is that it is having thread-like structures called as hyphae. The thread-like structures are called as hyphae. And from the hyphae, small blob-like structures, you can see blob-like structures which is attached to the tip of the hyphae. And these blob-like structures are called as sporangia. And inside this sporangia, it bears the reproductive units called as spores. These spores which are developed inside sporangia, they are lightweight and they are highly thick-walled. And once the sporangia matures, it bursts open. It bursts open, letting out all the spores out. All the spores comes out. And it being lightweight, it is carried by the wind. And once it arrives on a suitable surface, which allows or which provides suitable conditions for it to germinate, it germinates into a new hyphae. So, dear children, we have come to an end of asexual reproduction, the type of reproduction in which there is no fusion of gametes, only a single parent is involved, and the offsprings produced are exactly similar to their parents. The different types of asexual reproduction we studied are fission, fragmentation, regeneration, budding, vegetative propagation, tissue culture, and spore formation. Thank you.